Excellent. Yeah, thanks a lot for the introduction, Lenore. And also, I'm delighted to be here. So yeah, as Lenore said, my talk will cover today the real-time analysis of raw nanopore signal analysis for genome analysis.
Did you start recording?
Yes, yeah. Again, maybe quick self-introduction of myself. I'm John. I'm a PhD student, senior PhD student in the software research group at ETH Zurich. My research interests mainly lie in the intersection of designing algorithms for bioinformatics and also designing hardware, again, for bioinformatics. And I'm mainly interested in developing hardware and software for real-time genome analysis and also for sequence analysis and also for genome editing and error correction. So you can get to know us and our group using these websites. And I already include the QR code in the first slide. So these slides are already available online on my website. So you can go and find all of these links that I'm going to be showing you. And our group is led by Professor Onur Mutlu. He's a full professor at ETH Zurich. And he's currently also doing his sabbatical at Stanford University. And our group is a software research group. We are around 40 plus researchers in the group. And we work on many different topics such as computer architecture, hardware, software co-design, systems, bioinformatics, security, and memory, and so on. And our motto is think big and aim high. We have 4 key directions in our group, but today I'm going to be talking about designing algorithms and architectures for AI, ML, genomics, and medicine. So let me first give you some overview of the research that we're doing in our group, and then I'm going to move on to the main topic of today's talk. So, and I usually like to start with this quote from Richard Hamming, which is basically the purpose of computing is to gain insights, not numbers. Scientifically speaking, let's say, if it is not for entertainment purposes. And today, actually, we need to gain insights and observations much more efficiently than ever before. And the reason for that is basically we're generating big data. We're generating data everywhere in like many platforms such as social media, genomics, astronomy for scientific purposes. And basically the speed that we're generating data is currently far more outpacing the speed that we can process this data. And the reason for that, the machines that we're using to generate this data is really specialized at generating data at a very high throughput. So they are really good at generating data at high speed, but we're trying to compute this data using the general purpose machines, which usually don't have, let's say, any idea about the pattern structure or the needs of the data. And because the purpose of these machines is basically one size fits all, right? So, and when we look at basically the main problems in the general purpose computing machines today, we see that the data actually needs to move from the initial place that is generated, which is in our case a sequencing machine, right? And then it is usually stored in the storage, for example, SSD, HDD, and then it moves to memory, and then it moves to the CPU. And then here, there's basically a huge amount of data movement spent across all these levels in the computer architecture. And when you basically look at the energy and the latency that we're spending to move the data up until the moment that we compute the data, we see that actually the energy that we spent is around more than actually 100x more expensive than computing the data. So there's a huge, basically, data movement bottleneck in terms of both energy and also the performance. And so based on this observation, we clearly see that we need intelligent algorithms and also intelligent architectures that handle data well. So the main question is, given the needs of the data that we want to analyze, what sort of set of architectures we should use. There are commodity hardware such as FPGAs, GPUs, CPUs that many people are using, but there are also emerging technologies that people are looking at. For example, now we can use the DRAM itself or the SSD itself to do the computation. So you don't really have to move the data from SSD, from memory, and then based on, of course, the limitations of the hardware that you're using and based on the needs of the application, you can essentially decide to do some of the computation in these emerging technologies. And basically, this is an important question because there may also be special needs, such as we may soon be able to do portable sequencing, right? In-field sequencing and in-field genome analysis. And in such a case that certain conditions, for example, the battery limitations or computational power limitations may play an important role there. So the question is, how can we enable, let's say, portable, energy efficient and fast computation, especially for in mobile devices? So we look at all these, essentially the entire stack of computer architecture, and then look at what steps in genome analysis need a special focus for hardware software co-design. And then we've written this positioning paper last year. This was an invited paper. So if you are curious about basically what the opportunities are for co-designing hardware and software together, you can take a look at this paper. And as I said, we're mainly looking at the entire stack of computer architecture, starting from algorithm up until to the device level, and then trying to co-design everything together. And basically, the applications are only limited by our imagination when we consider all these levels. Especially, I'm interested in genome editing. And also, DNA computing is very interesting application, I would say, for particular reasons. And also, we're looking at these newer genome sequencing technologies, relatively newer. Although nanopore sequencing has been around for some time, but the opportunities, I guess, now are revealing a lot more. So we look at these new genome sequencing technologies, and we also work on this direction. So this is one paper that I'm going to be presenting today, RowHash. Essentially, we've published several works in this direction. This is RowHash 2. And I also mentioned a couple of students here, RowAlign and also Target Call. That also deals with the raw nanopore signal analysis. But we also look at the sequence analysis level. For example, how can we achieve fast inexact match, inexact seed match without using expensive, let's say, LSH mechanisms. And also we look at essentially the question for liftover, like in case like your reference genome is updated, do you really need to do your entire read mapping from scratch, or can we move the existing information efficiently so that your computational bottleneck is reduced? And we also use ML, let's say, for error correction purposes. This was published a few years ago in bioinformatics for correcting the errors in assembly. And then we also use ML and genome graphs for executing certain tasks using profile hidden Markov models, specifically for error correction purposes, but also for multiple sequence alignment and so on. And then we co-design algorithm and software together. This paper was published in ASM Taco just a few months ago. And also, of course, like again, we co-design particular software and hardware. For example, this is for approximate string matching. And this is again for sequence to graph alignment. So all these papers are published and presented in top computer architecture conferences. So we also design filters in the SSD and ask the question which sort of data we need to move from SSD to memory to do the read mapping operations. And turns out that we can actually figure out the exact matches much quickly in SSD, where you don't really have to move the data for such cases, and you can report exact matching alignment using the operations in SSD. And also, we use processing in memory in order not to move the data again to CPU, but rather do the operations, computations in the memory as well. And then this paper deals with the base calling plus read mapping problem. And then this is another paper that performs base calling again using processing in memory. So these are basically the scope of the works that we're doing in our group. And today I'm going to be focusing on this enabling fast and accurate real-time analysis, analyzing the raw nanopore signal data. And I'm going to mainly be presenting RowHash and RowHash 2 papers. So RowHash was presented at ISMB last year. So you can use these QR codes to access the paper and the source code. So let me give you a little bit background then before I start describing RowHash. So when we look at the raw data that these sequencing machines generating, so they all generate different kind of data, right? But they all generate raw sequencing data at one point before they are converted to the base called sequences. And today we're going to be mainly looking at this nanopore sequencing technology, which generates raw electrical signals. And I'll try to give the reasons for that. But mainly, the nanopore sequencing is a widely used sequencing technology in our field. So it can produce long reads up to 2 million nucleotides. It provides opportunities for portable sequencing because there are these devices that can literally fit in your palm, so you can carry it with you and then do your portable analysis. It is relatively cost effective. And there are also some more unique features, such as real-time analysis, that I'm going to be covering now. So if you're not familiar with how nanopore sequencing works, so you can imagine a 3-layer solution here. I'm not sure which part is maybe it's easier. So you can imagine a 3-layer solution here. So on the top layer, we have a negatively charged region. And on the bottom layer, we have a positively charged region. And in the middle, we have a particle membrane where this pore that we call nanopore is sitting. So what's happening here is that since the DNA is also negatively charged, so this DNA is literally moving from the negatively charged side to the positively charged side. And as it moves through, it creates ionic current disruptions over there. And these disruptions are measured and reported as electrical signals. And these disruptions are significant enough for us to tell each base, each nucleotide in this DNA molecule, so that these electrical signals are then analyzed. Let's say we could be translating these electrical signals to ATGCs again, using a computational approach called base calling, or we could also directly analyze these raw electrical signals without base calling them. But the opportunity here is that these electrical signals are reported in real time. So this means that we can also start analyzing these electrical signals in real time while they are being generated. And one very important feature of nanopore sequencing is, based on your analysis, you can also give feedback to the sequencer and telling, for example, if the read that you're currently sequencing is somewhat useless to you, or basically you've done enough analysis for that particular read, you can tell the sequencer to stop sequencing that read at runtime. So this is basically very advantageous, and I'm going to be describing those benefits now. So there are 2 main benefits with real-time analysis. One benefit is, in your most common case, what you would do, you would first sequence, get your data, and then do your analysis, right? And then this is the entire time you would spend. But with real-time analysis, you can sort of overlap the sequencing time with the analysis time. And then this would basically reduce the overall latency of genome analysis. So this is one advantage. The second advantage is that, again, in the usual case, we would be completely sequencing reads. So you don't have the option to tell the sequencer, oh, I want to stop sequencing that read in the middle, let's say, so that you can save maybe from sequencing time and cost. But with real-time and nanopore sequencing analysis, we can do that. We can partially sequence the read. So this means that we can reduce the sequencing time a lot, and also potentially the cost by doing so. But of course, there are challenges for real-time analysis. One challenge is, of course, we need to match the data generation speed. Our computational approach should match the data generation speed. Otherwise, it's not real-time. The second challenge is that we really want to make these rejection decisions or ejection decisions extremely quickly, so that we don't sequence unnecessarily, basically. Of course, also we also want to do accurate analysis, right? Our analysis needs to be accurate, that's obvious. And also, for portable sequencing reasons, we want our analysis to be power efficient, right? That's another challenge that we need to keep in mind. And so I already told you that we can perform raw signal analysis without base calling, and this is, again, a quick background on this. So you can here assume that at a time within this nanopore, there are k many nucleotides basically included within this pore. So this k many nucleotides is usually from 6 to 9 mer, depending on the technology that you're using, depending on the chemistry that you're using. And so this is basically referring to a k-mer, right? So, and we also have this term that we call event. So this is basically this electrical signal that is generated from nanopore sequencing. And this event corresponds to a particular segment in the raw signal. So what this event is basically the the part of the raw signal that we generated while we're sequencing that particular k-mer over there. So the events are basically corresponding to the k-mers, the sequencing of k-mers. And then whenever basically a new base is coming in to this nanopore and another one is coming out, there's usually an abrupt single change in the signal that is reported. And usually statistical methods are used to identify these abrupt changes. And basically then based on these markings or the abrupt changes that we find using statistical methods, we calculate the average, let's say the signal value over there, which then is referred as the event value. That corresponds to basically the signal value that we would have when sequencing a particular k-mer. But there is one critical observation here, which is when we're sequencing identical k-mers, we don't generate exactly the same signal, but we generate similar signals, let's say. So that's an important observation that we're going to use later on. So with that, I'll then start describing RowHash. So the problem that we see is the real-time analysis of raw nanopore signals is either inefficient or inaccurate, especially for large genomes. And our goal with RowHash was to enable this fast and accurate analysis in real time, specifically also for large genomes. And we make two key contributions in RowHash. So RowHash provides the first hash-based search mechanism for mapping raw signals, for mapping raw nanopore signals. And the second is we introduce a method that we call the sequence central technique, which can dynamically stop the entire sequencing run, basically not a sequencing of a single read, but the entire run of sequencing if basically further sequencing is unnecessary for the type of analysis that you're using. And we look at relative abundance estimation for that analysis, and I'm going to be talking about this more in the coming slides. And based on basically across 3 use cases and different data sets from varying sizes, including a human genome, we find that our work essentially outperforms the existing state-of-the-art works by 27x, 19x, and 4x in terms of the throughput, computational throughput. And also RowHash provides the most accurate raw signal mapping for all data sets. And when we look at basically the benefits of the sequence central technique, we also see that we can reduce the sequencing time and cost by 15x, basically, when this technique is enabled. So there are 2 solutions for performing real-time analysis. One, as I said, we could be doing base calling, and then you could be analyzing your base call data as you would do in real time. So this is good because it is usually a more accurate approach, right? This is considered as the golden standard for the best accuracy. But this is also extremely costly and power hungry. So you are usually using basically these expensive deep neural networks with your GPU and so on. This is basically limiting your scalability. So the second approach is mapping signals to the reference genomes without base calling them. And this is basically is beneficial for 2 reasons. Raw signals contain richer information than just bases. You can be inferring the methylation information, for example, as well directly from raw signals. And then it also provides efficient analysis with better scalability and portability. So I'll skip this slide because I already covered this one. But basically, when we look at the problem in these approaches that perform the mapping of raw signals, it's actually about the scalability for larger genomes. So when you have your raw signal, and then when you're mapping to a small region, small reference genome, what you usually have is that you have fewer candidate regions, right, in small genomes. So this means that you can somehow do some accurate mapping, and also you can provide high throughput because the problem size is not really large. But when you basically start dealing with the larger genomes, for example, a human genome, then this means that you have substantial larger number of regions that you need to check per read, basically. So the cost of computation per read increases substantially with the larger genome. So this leads to 2 problems. One problem is there are probabilistic approaches called uncalled. And when you're working with many regions, specifically with repetitive regions, this particular work cannot perform accurate mapping because it cannot decide accurately from these too many regions. So there is another work that's called SIGMAP. It performs distance calculation to identify the similarities and to perform mapping. But again, when you're working with a larger, let's say, reference genome, that sort of distance calculation becomes too costly again, so that the analysis itself stops becoming real time. So then we find that the existing solutions are either inaccurate or inefficient for large genomes. And our goal is to essentially solve this problem by enabling fast and accurate analysis. So to this end, we propose RowHash. We make two key contributions. We provide the first hash-based search mechanism, and also we propose the sequence until technique. So I'll first describe the hash-based search mechanism that we provide in RowHash. So remember the observation that I already mentioned earlier, right? So when you have, let's say, when you're sequencing the identical nucleotides, assume that you're sequencing a particular k-mer twice, right? So what's going to happen is that you're going to generate, let's say, two different signals, and these signals are not going to be exactly matching. But the observation is that these signals are going to be similar to each other. And the earlier work exploits this observation by calculating the distance between signals. And when you calculate distance, you can tell the similarity and so on. But we don't want to do this because it is costly. Rather, what we want to do, we want to somehow generate hash values from these signals. And these hash values then can be used for fast match purposes, right? But then the question is, there are basically two challenges. And one question is, how can we generate the same hash value from different signals or similar enough signals, right? So we need to be able to do that. The second challenge is that we also want to accurately find as few similar regions as possible, right? So that we're not really looking at too many regions at a time, but just only a few regions, even if it is a large genome. So this is the overview of RowHash. So we start with converting the reference genome into signal space as well in our indexing. And similarly, again, we convert the raw electrical signals to the event values that I described you earlier. So we complete the work on the signal space, including the reference genome. And then to eliminate some noises, we perform quantization, binning, and so on. And then we generate hash values from these noise-free, almost noise-free values, so that we can store these hash values in the hash table for reference genome. And then later, when we're generating the hash values from the reads, we can create the hash table so that we can do our usual chaining and mapping steps that we do in Minimap2, for example, right? And the steps here on the left is an offline step, is just a one-time task for each reference genome. And the right step, the right step would be here, the real-time analysis part. So I'll start with the reference to and also signal to event conversion. As I told you, we're converting the reference genomes to signals, right? And the way that we're doing it is essentially we essentially take the k-mers from the reference genome, all overlapping k-mers from the reference genome. But the size of the K is really important here because it is really dependent on the nanopore sequencing, nanopore chemistry that you're using. So remember, I told you, like, usually it contains the 6 mers or 9 mers within a nanopore. So you should be basically taking the correct k-mer size here when you're converting your reference genome to signal. And then what we do is that there are these lookup tables that tells us, given a k-mer, what is basically, so these k-mer models are pre-constructed based on the nanopore, basically the characteristics of the nanopore. But what happens is that given a k-mer, it tells us what is its expected signal over here. And then we can do this for all k-mers and then do normalization and so on, such that rather than basically working with k-mers, we work with the signal values of these k-mers. So this is basically pretty simple. This is a one-to-one conversion of the k-mers to the signals. Then we also convert, let's say, the raw signals to events, because we just converted the reference genome again to events from their k-mers. And to do this, what we simply do is that given a signal, we perform this segmentation, which is a statistical test that identifies these abrupt changes in the signal, which is our events. And then we calculated mean and normalization and so on. But the values that we're getting at the end is somewhat comparable to the values that we generated from the reference genome. So now that we have these basically consecutive events, which are supposed to be consecutive k-mers, so the question is, can we directly match these signals, or can we directly match these events to each other to identify similarities? So for that, I'll cover the quantization step in our work. Again, I'll remind you the observation, which is basically that there are slight differences in raw signals, right, from identical k-mers. So this means that when you're sequencing this k-mer, you may end up generating different values, but they are going to be similar to each other. So you cannot simply try to match these values to each other, because you're not going to be able to match them, right, with a quick search. Rather, what we do is that we simply quantize or bucket or bin these values. There are different terms for that, to essentially reduce the value space, maybe from like 1,000 different values to around 32 different values, because here we use 5 bits over there. But the idea is basically binning over there, right? So that basically these values that are closer to each other can be binned into the same values, so that we can reduce the noise effect from these signals. So this is quantization. And then the last step would be generating the hash value. But there is one more challenge that we need to solve, which is basically about the length of these events, right? So when you try to match a 6-mer in the reference genome, you're going to find maybe thousands or millions of matches, right, in the reference genome. But we just quantize that k-mer, so we cannot simply use this 5-bit value or this particular small value just for matching purposes. So we need to somehow increase that k-mer size, basically. And the way that we do it is we assume that the consecutive events are consecutive k-mers. So we can somehow preserve the order information by essentially packing all these different quantized values together. Maybe we may have like 5 or 6 quantized values here. And then when you pack them together, what you end up getting is basically a huge and distinct enough hash value, which can be used for matching purposes, because this is already long enough and unique enough, hopefully. So this is basically how we're generating the hash value from raw signals. And then to basically wrap up all the steps that we do for real-time analysis, here we construct the hash value from the reference genome by converting it to signal. And then what we do is that we store these hash values and their positions in the hash table, right? This is indexing. And then again, in real time, so this is stored. And then in real time, what happens is that we similarly generate the hash value again from raw signals while the raw signals are being generated. And then we query the hash table. And if there are some matching positions, we perform chaining and mapping and so on. And based on the mapping information that we generate, we ask the question continuously whether we should keep mapping, meaning whether we should keep sequencing this read or not. And if the answer is yes, we simply retrieve the next chunk of data, do all of these steps again, and then do this checking again, and then so on. But if the answer is somehow no, then we tell the sequencer to stop sequencing that particular read using the features that are known as read until or run until. Basically, this is the entire real-time analysis using RowHash. So let me see how I'm doing with time. OK, I guess I have a little bit more time to cover the sequence until mechanism. So the sequence until mechanism is somewhat different than RowHash, but there's a good synergy, let's say, between two, because they can be used together for reducing the sequencing time and cost. So the problem that we're dealing here is that unnecessary sequencing obviously wastes time, power, and money, and we don't want that, right? And the key idea in sequence until is to dynamically decide if further sequencing of the entire sample is necessary to achieve high accuracy, right? That's the key question and the idea that we're trying to answer. And then if it is not necessary, we can stop the sequencing early without sacrificing the accuracy. And the potential benefits, of course, the significant reduction in sequencing time and cost. And then for the analysis purposes, we look at a particular use case, which is a relative abundance estimation. And then we try to perform relative abundance estimation without sequencing the entire sample. And there are basically a few key steps to achieve this. So what we do is that we continuously generate relative abundance estimation after sequencing of every n reads. For example, we just sequenced I know 50 reads, and then based on that 50 reads, we do some relative abundance estimation, right? Now this is your estimation. And then we basically keep doing this, and then we always keep the last t estimations that we had previously, right? So this is the last t estimations as we keep sequencing. And then what we do is that as soon as we have t estimations, or as soon as we have a window of t estimations, we ask the question whether there's an outlier in our estimations. So if there's an absence of an outlier, then this means that we are performing relative abundance estimation pretty consistently. So this means that the further sequencing is likely also to generate the consistent results. So then this means that we don't really have to keep sequencing the entire sample. Rather, we can stop the entire sequencing run, because when we looked at the past window of t estimations, our estimations basically didn't change much. So we have a pretty good confidence that it won't change with the further sequencing as well. And basically, if there's an outlier, then this means that the sequencing keeps changing the results substantially. So this means that we just need to keep sequencing it, and then we do it basically until we reach to this decision point. So this is sequence until, basically, and then we can somehow merge these two with RowHash and RowHash2. With just a single or two slides, I'll cover the optimizations that we make in RowHash2. So RowHash2 is a follow-up work on RowHash to improve its accuracy and performance. And what we bring up in RowHash2 is basically we have this new quantization mechanism that is more accurate and also more efficient. We also improve the chaining algorithm with more sensitive penalty scores. Our decision making for stopping the mapping process is now like a ML decision making. So we have this almost single layer, single neuron architecture over there that makes a more robust decisions. And also we integrate filters and minimizer sketching as well to achieve more space efficient, and also to reduce the seed matches for our analysis. And then if you want to basically imagine how the minimizer sketching could be integrated here, is basically when you have your hash value over here, what you could simply do is that now that we are in the hash value space, you could literally use all the existing sketching techniques out there, right? Strobmers, minimizers, or like any other sketching technique, and then sample your hash value based on that, and then generate your index accordingly, and so on. And then in the RowHash2, we integrate minimizers for that purpose. So I'll then quickly cover the evaluation. For RowHash2, we have two settings. We have RowHash2, the default setting. We have all the k-mers, and we also have RowHash2 minimizer with minimizer sketching. We compare to the state-of-the-art works, Uncode, SIGMAP, and our early work, RowHash. We have three use cases: read mapping, relative abundance estimation, and contamination analysis. And we have three evaluation metrics. One is throughput, which measures the basis process per second per CPU thread. We also look at the potential reduction in sequencing time and cost of each tool. And also we look at accuracy. And for accuracy, our baseline is the mapping of base code reads using Minimap2. And we look at the precision recall and F1 scores for accuracy. And for relative abundance estimation, we calculate the distance to the ground truth again, which is generated using Minimap2. We have 5 different data sets for read mapping, starting from a very small viral genome up until the human genome. For relative abundance estimation, we create a mock community by combining all of these data sets. And for contamination analysis, our sample includes the viral genome and also the human genome. And the question is basically whether the sample is contaminated by a viral genome or not. So we will first look at the throughput results, but I'll again remind you, the real-time analysis requires faster throughput, faster computation than the data generation speed, right? And the throughput that we get from a single pore is usually around 450 base pairs per second, right? This is the data generation speed per nanopore. And here we show the results for each tool. On the y-axis, we have the throughput numbers over here, and on the x-axis, we have the data sets and the use cases. And based on these results, we make two key observations. One observation is RowHash outperforms, improves the performance of throughput basically substantially compared to all these state-of-the-art works, including RowHash by 4x, its predecessor. And also, when we look at only the performance of RowHash 2 minimizer, it further improves the throughput results by 2.5 x compared to RowHash 2, because minimizer is a sampling mechanism. You have, let's say, a smaller problem to work on. So we also look at the average sequencing length, average sequence length when you're using each of these tools. And basically, here we want to achieve fewer bases. We want to sequence fewer bases, because then this means that less unnecessary sequencing. And here on this plot, the lower is better. And on the y-axis, we have the average, the sequence length that each tool requires before stopping the mapping or stopping the sequencing. And again, on the x-axis, we have the data sets. So we see that the RowHash 2 reduces the sequencing time and cost on average by 1.9x compared to Uncold and RowHash. And also, RowHash leads to the sequencing the least amount of bases for larger genomes. And what I mean by larger genomes is these ones except the viral and the E. coli. And for E. coli and viral, Uncold seems to be doing better than RowHash 2. And last, basically, when we look at the accuracy of read mapping, so what we're doing, we're mapping the reads to their coordinates in the chromosome, and we're literally looking at whether these mapping positions are correct or not compared to the min-map2 positions, right? And then for that, we have this, we measure the F1 score for read mapping, we measure the precision for contamination, and then we measure the distance for relative abundance estimation. And the observation that we make from these accuracy results is RowHash provides the most accurate read mapping for all data sets and for all use cases. And so I told you that RowHash 2 is fast. The question is it accurate? So when we look at the results, RowHash 2 minimizer provides an on-par accuracy with RowHash 2, but with, of course, substantial throughput benefits. So if you want to trade off between performance and accuracy, you have already your two answers over there. So I guess one last metric that I want to show is about the benefits of sequence sample, right? So in my opinion, this can be applied to all other tools other than RowHash, and we also have simulated results for this. And what we do with this evaluation is essentially we let the RowHash run by using the entire sample without the sequence sample capabilities, right? And then we make a relative abundance estimation using the entire sample. And then we activate the sequence sample mechanism, and then let the mechanism decide when to stop sequencing the entire run, depending on the outlier detection that I just told you. And then when we do this, we find that sequence sample stops after sequencing the 7% of the entire sample, right? So you don't know this number beforehand. You only know it after you do your analysis. And then after sequencing 7% of the sample, when we look at the distance of our estimation compared to the distance when using the entire sample, we look at the distance, we find that the distance is basically pretty low. So you can almost achieve the identical accuracy by only sequencing 7% of the entire sample. So this means that the sequence sample enables sequencing only using the 7% of the entire sample with high accuracy. And then we also simulate the benefits of sequence sample using Uncold, not just RowHash. We basically look at certain breakpoints, like what would be our relative abundance estimation at 1%, 10%, and so on. And then we find that actually we can reduce the sequencing time and cost by up to 100x using this sequence sample mechanism. So to conclude, RowHash provides the first hash-based search mechanism that is critical for fast and accurate analysis, especially for larger genomes. And the sequence sample can dynamically decide whether further sequencing is necessary for accurate analysis. We show, based on our analysis on different data sets and use cases, we find that RowHash is much faster than the earlier works. It's the most accurate mapping tool, and the sequence sample reduces the sequencing time and cost. And we essentially, to wrap up and to some, I guess, give motivational points here, now that we can work in the hash value space, so this means that we can bring all the existing works in the base code domain that uses these hash values to perform certain analysis, right? We can bring all these analysis now to the raw nanopore signal space. And we also know that the indexing is very cheap. So this means that there are many future use cases that can be performed with on-the-fly index construction, perhaps. And so all this means that I believe we should rethink the algorithms to perform downstream analysis fully using raw signals, right? I don't see currently a reason why we shouldn't do or why we cannot do all the downstream analysis fully using downstream analysis. Maybe in the future, many tools will be developed for this purpose. So this was RowHash, and I'll quickly conclude again using a few other slides. So one of my conclusion remarks is I believe future is bright for raw signal analysis. So this work is not public yet, but it's going to be public soon. And one reason why I think it is bright, because now using these efficient hash-based search mechanisms, we can also perform overlapping in real time. And I think you already know what this means. So we can hopefully construct assemblies also in real time. In fact, we basically try to do it with the on-the-fly overlap construction. First, we look at the quality, or basically how many overlaps that we're sharing with the base code overlaps, right, using all these 3 data sets. And especially for smaller genomes, we see that the raw signal overlapping shares a large portion of it with the base code overlaps. But still, there are some unique overlaps that are unique to raw signal overlapping. And also there are some overlaps that are unique to base code overlapping, right? Then the question is like, are these unique overlaps perhaps more accurate, or like since we're not finding these unique overlaps in Minimap2, are we reducing the quality of or contiguity of the de novo assembly? And then what we do is that after constructing the overlaps, we also construct the assembly from these raw signal overlaps. And then we currently look at the contiguity of assemblies, and then we see that the answer for contiguity for almost all data sets is yes. The signal-based overlapping is currently less contiguous, but it gives some, I guess, exciting results, because in terms of the average contig length and the maximum contig length, we are already pretty close to, almost close to basically the base code overlapping. And we're basically just starting, I would say. And when we look at the human genome, actually, the surprising thing is, this is a very, let's say, low sequencing depth of coverage. What we observe is that with a low coverage, we can actually construct more contiguous and longer contigs using raw signals than base code. And we don't know the answer to that yet. So we'll look into it. We'll try to understand, perhaps maybe there are richer information. There's richer information in raw signal that leads to more contiguous assembly when the sequencing depth of coverage is low. There are basically new applications that we can keep working on, right? Especially with the overlapping information. So you could imagine, you could use the overlapping information for improving the base calling accuracy, right? You could give your base caller this overlapping information, and it could utilize it, right? And as I told you, I already told you that we can also perform full downstream analysis, fully using raw nanopore signals. I think that there's many work that can be done in this direction. And then we could also cooperate the raw signal analysis with base calling analysis, right, together, if this is useful. And also there are many, many more, I guess, exciting times and applications, keeping the hardware design in mind. And speaking of the hardware design, I basically tell the things are also happening in industry. So I'm pretty sure maybe you're already aware of this, the Dragon platform that Illumina provides. This is a full FPGA board that performs your entire genome analysis from mapping to variant calling in a faster way, right? And these are already included in your Nova and Nexec Illumina devices, right? So here they are basically also motivating the hardware acceleration and also the use of FPGAs for improving the speed of genome analysis. And there are also undergrad startups that simply basically exploit the GPUs and then accelerate. A very important application here is G8K. And then they accelerated so much that the NVIDIA actually buy them out, let's say. With simple ideas, you can even maybe perhaps earn millions of dollars, I assume. And also the NVIDIA and also other vendors are essentially looking at these important applications. Genomics is one of them. And then looking at what sort of operations are needed for these important applications, right? And dynamic programming calculation is one of them. And now the dynamic programming operations are now also natively supported in some of the GPUs, right, to accelerate certain applications, including perhaps read alignment and chaining. And also, certain genomics companies are also collaborating with the tech companies, such as, for example, here, we know that Bionono is collaborating with NVIDIA to accelerate their analysis. And now people are also looking at how can we basically design hardware from scratch to accelerate certain applications. Here, this is a Cerebrus wafer scale engine. This is an AI engine. It includes 2.6 trillions of transistors to accelerate AI ML operations. And then there are now also in-memory devices. So these are DRAMs that you can buy off the shelf, but they also have the capability of computing as well as basically storing the data. This is UpMem, and you can buy their modules and then do your analysis within the memory. And we are also a part of basically the effort that we call BioPIM, where we try to basically understand certain genomics applications and then try to see the opportunities, which of these applications we can use for in-memory computation, right? So which algorithms can we bring them to the in-memory space? And there are basically many partners in this group. There's Bilkent, there's IBM, ETH also, also some groups from France and so on. So we also publish and make certain materials available. So in case you want to learn more about basically the use of hardware and software together for fast genome analysis. So if you want to learn more about this, you can take a look at all these materials, and these slides will be available. Is already available on my website. We also organize workshops in this direction. Last year at Reconf, we organized the BioArch workshop where we had talks in this direction. And we also have courses, again, in this direction. Many students take these courses and then learn how to use hardware for accelerating, to accelerate the genome analysis. So in conclusion, so today we covered various recent ideas to accelerate genome analysis and also to analyze the genomes in ways that were not possible before. And in my opinion, enabling cost-effective, portable, fast, and accurate genome analysis has many implications. So basically, for example, what are the new applications to enable these unique benefits of, for example, raw signal analysis, right? And maybe next question is, of course, can we do even better? Can we sequence other types of biological data, maybe even amino acids, and then perform, basically do proteomics also in real time using raw signal data? And basically, to conclude, I think many future opportunities exist, especially with the new sequencing technologies and also especially with the new applications and use cases using the emerging hardware. So with that, I'll conclude my talk, and I hope I can get some questions without running out of time. Thanks a lot.
Wonderful talk. Thank you so much. I have a couple of questions, but I won't dominate. I'll do one and then I'll come back. You too, but I want to. Yeah. So my first question is an algorithms question. So there are, when you're aligning to a reference genome, and because of personalized genomics, you're going to see a lot of SNPs, right? Mm-hmm. So my question is, for your hash values, have you done anything to hash things so that nearby k-mers hash to similar values? There's this locally consistent hashing. Are you looking at this now? Or are you smiling?
We have a paper about this blend. I can talk about a little bit, but maybe I'll just you can finish your question first so that I can answer. That's all. Okay.
Looking at versions of the hashing that, if your k-mer is off by 1, you hash to a similar value.
Excellent question. This is actually an ongoing work. Currently, this is only exact hash value matching, say in RowHash and RowHash2, although we have some quantization. The purpose of quantization is to remove the noise from the same k-mer. Yes.
Is the noise that you get in the values really stochastic, or is it context dependent? Can you get different range of values if you like TTT versus like DAT, or is it?
Yeah, it is both. So there is stochasticity definitely because of the physical environment, how the sequencing is happening. Even the temperature is affecting the range of these values that you're getting. The quality of the pore is also affecting, right? Over time, it ages. And there are residues also in the nanopore, which is also affecting them, which is something that you cannot control. This is the stochastic part. But we also look at whether it is all stochastic, right? We also see a particular pattern where certain base, so the signal that we're generating is for a particular k-mer, right? But when we look at it more closely, what we see is that usually the fourth or the fifth base of that k-mer is affecting the signal much more than any other base in the k-mer. The reason for that is probably because it is the closest base that is closest to the header of the nanopore. So it is giving the most disruption in the ionic, basically in the current flow over there. The other ones that are close, that are nearby, still disrupting the currents, but not as much the one that is closest, that the header, where the header is located. So there is that pattern as well. So it is both, I would say. For the first question, I already told you that it's exact matching based, but we have this work that we call blends, which I presented last year at Recomp also. The question over there, given k-mer or a sequence of a certain length, right? Can we generate the same hash value if a pair of sequences are similar enough to each other? There are works that do that, for example, space series, right? But they insert basically masking positions at fixed locations, right? But the I believe so, yes. Yeah. Exactly. So usually the challenge over there, you either have too many hash values that you need to generate, and then you calculate Jacquard similarity, or one of the hash values should match, right? But the real question over there, can we actually generate a single hash value that enables the mismatches at any arbitrary position between a pair of sequences, but we can still generate the same hash value, single hash value, so that we can just use it as if we're using it for exact matching purposes.
Okay, so no, so that's not the problem they're solving. They're doing nearby. Just, but then you can quantize the same as you're doing for the exact hash, right?
Right.
If I was able to give you a range, and whenever the k-mer was close enough in Hamming distance, I was also not too far from that range, and you could also have a quantized signal where you could do what you're doing.
Right. Yeah. We also use the nearby k-mers, but I guess maybe for different purposes in blend. So it's using sim hash technique.
So new minimizer technology, and yep, there's a whole step.
Exactly. Exactly. Yeah.
I'm not so familiar with this topic, but I thought that, yeah. In fact, I was, you mentioned that you are trying to hash things into one single fact instead of like multiple, facts.
Could you elaborate more? What do you mean by single?
I saw that perhaps through what you were describing, but when you were answering the question, you were saying you're actually one single.
Exactly.
So how did that actually one single? Like, well, maybe let's go back. How large is the entire search space?
If for the entire reference genome, I guess you're referring to, or for the number of bits that we're allocating for hash values. For hash values, our search space is 2 to the 32. We use 32 bits, so entire search space is 2 to the 32. And when you're quantizing, you usually have 5 bits. But what you can do is that you can actually pack them together next to each other, right? It's like some bit manipulation. You can use the most significant 5 bits for the first k-mer and the next for the next k-mer and so on. So you can create these large, maybe even 60-bit value, maybe merging, I don't know, 12 events. And then you can give the 60-bit value to your hash function to compress it to the 32-bit value. Yeah.
I'm curious, are you working like with Oxford Nanopore to put this on their devices, or like anyone who owns a device can like put your algorithm in there?
Yeah, so we're not working with them currently actively because they already have a platform where they enable base calling in real time. They don't really do raw signal analysis over there, but they provide the API for us to do. And actually the API is almost integrated there. So soon in RowHash, what you're going to be able to is actually you're going to be able to connect to the Nanopore device and then run RowHash directly connected to the Nanopore device using that API. But we're not directly working with them at the moment.
I have one more question. So yeah, RowHash does the mapping to the reference genome. Can it do like just base calling instead? And if it can, like how the quality would compare with like Dorado or like Nanopore real-time base calling, like on GridION or something like that?
Yeah. So base calling, given the right setup, base calling would be more accurate. But what I mean by right setup is the following. Base calling usually requires a large chunk of signals, which is usually larger than a chunk of signal that you receive within 1 second. It's usually, I guess, like around 2 to 3 seconds, right? Unless you get this, the base calling accuracy is actually pretty low. And there are works that show that the accuracy would be extremely low compared to these directly in mapping approaches. So, but then the next question is, if you're getting a large chunk of data, assume you're getting a 3 seconds of data, right? Then you're basically bounded by making your earliest decision at the 3rd second, right? The question is, maybe you could do your decision much earlier, right? Maybe in the 1st second or in the 2nd second, right? And this is important because after some point, for example, if you sequence for 5 seconds or 6 seconds, right, the DNA that is already sequenced grows a lot on the bottom part of Nanopore, such that when you decide to eject it, so ejection process is basically you switch the voltage such that the top side becomes positive charge and the bottom side becomes negative charge. So your DNA starts moving to the opposite direction, right? So it sort of is ejected from Nanopore. But if the DNA that you sequence so far is long, then if you make your decision late, that DNA, there's a high chance that will be tangled in the pore. So you're going to block your pore basically if you make a decision late. So that's another challenge, right? So you really don't, I didn't talk about this basically in my talk, but you also really want to make quick decisions. So you could configure to make it quick decisions with base calling, but then your accuracy will be lower than base calling. So it's like a two-way approach, and depending on your needs.
So for example, if I run it offline, not in real time, then like the quality would be comparable to Dorado?
Yes, I would say. And we already have, so those mapping results that I already showed you is based on compared to the base calling. So this is basically the ideal setup, right? So when you, for example, look at the F1 score 0.98, let's say, take the most accurate one. This is telling you that actually the mapping information that you're generating is almost identical to the mapping with the base called approach. Yet still we're using smaller information, right? Just a part of the data. While this mapping information, the base called mapping information, uses the entire read information, right? If you had used the entire read information, I'm pretty sure those accuracy numbers would be even much higher. So that's why I'm pretty sure that the accuracy would also be high when we did a similar approach with full reads.
That's cool. You have to commercialize this if it doesn't require the GPU.
Maybe, yeah.
So do you treat each event as independent in the hashing? Because it seems like the physics would have, like the previous event would still have like lingering.
Excellent question. That's a very good, insightful question, actually. That's true. I omitted that detail again in my talk, but when you have subsequent events where their values are close to each other, we only pick one of those, just because of the effect that you just told, because the segmentation algorithms are not perfect, first of all, right? It may not identify the events correctly. The second is there's this lingering effect that's going on, right? So then this means that if they are close to each other, there's a high chance that there's an error. So just let me pick the first one, and then the next one that I pick would be the one that has a certain difference between the one that I picked and the next one, basically. So we pick those so that they are different enough.
Without giving too much detail, can you explain a little? No, because it's a detailed question. Can you explain a little bit how you actually differentiate the signals? Like, you mentioned that when there's a significant drop or a significant change in the signal,
Right.
you categorize it as different from the previous signal.
Yeah.
And so on. How do you make that decision? How do you know that that is the spot where you will change?
So
the signal change?
that's a simple statistical t-test, also known as student's t-test. So you have maybe two windows, maybe you could have a single window as well, and then you continuously check whether basically a particular point is significantly different than, right? So that's the approach that we're taking, but it's not perfect. Maybe some ML approaches also could be used over there.
Because one quick version of this is that when you were talking about accuracy rates, so the human genome is harder, right? And
Right.
for many reasons, it's legitimately harder.
Right.
Have you been, I mean, you talked a little about the potential for integrating this with other approaches. Have you been doing a short and long read version or something else to get accuracy up on human or human scale problems? What else have you been doing? Or I don't know, maybe can you just talk about that number a little, because that's discouraging.
That's true. I agree with that. And so it is encouraging for me. The reason for that is we started from 55
Right.
for RowHash.
But that's not,
I agree.
yes, so it's better, but it's not.
Exactly. It's not in the ideal way. Yes. Exactly. So we did some empirical analysis to see what we can achieve with accuracy without really caring about performance, right? The speed. And we've basically played with certain parameters just to basically see whether our seed matching approach is limited by something, or if it is limited, like what is the limit over there? Now, what we've seen was the F1 score that we've seen the best was around 0.9 for human genome. But then the computation is pretty slow because of like either you have too many matches, you either have like too many chaining computations that you need to make. So to make it better, I think better filtering algorithms need to be developed, or even so that even if you have too many seed matches, specifically from repetitive regions, which I believe it is coming from there, right? So there needs to be basically better filtering algorithms that can distinguish or that can filter better the seed matches when we're hitting the repetitive regions. So I believe that's one thing. The second thing is, of course, we can still keep improving the quantization mechanism. The hash value that we're generating is still more accurate, so that, again, maybe some of these wrong matches in the repetitive regions are, again, eliminated. So that's the second approach that I think we could take.
I'm not sure if the hashing is going to help you with the repeats, right? Because they should hash at the same place. So the question is, but you have long reads, and the long reads should help you with repeats. So, but you're looking at little pieces of the long reads, which is throwing that away. So how can you sort of take the repetitiveness and say, wait a minute, we have something that's, you know, a megabase away. Can we use that?
That's true. Perhaps, yeah. Maybe fully using the long read information could be useful. Although we're doing chaining algorithm as well, right? It chains like several seed matches in the large region still. But still, I guess there's a high chance. Exactly. That's true. I agree
So
with
yeah,
that.
I feel like you're actually, you have the information and resolve a lot of that.
Exactly.
So
I completely agree with that. We definitely have the information. The question is the redundancy, definitely.
I
Right.
got you. It's not an easy problem, but it seems like.
Right.
we got that.
Exactly.
I'd also love to see fly up there, because something in between, something in size between yeast and, I don't know how big algae is.
It's 100 million bases, I guess, if I remember correctly.
Yeah, so fly is one more, yeah.
300.
Yeah.
No, let me see. Genome size, yeah, 100 base, 100 million, yeah.
So fly might be a good intermediary,
Yeah.
where it's
Oh,
still,
yeah.
it's not anywhere near as repetitive as human, but it's larger than,
Okay, that
I don't know.
that's true. Yeah, that's a good tip.
It's been
I agree.
another test bed. Cool.
That makes sense.
Good question. We can stop and take things offline. All right, let's think.
Okay, great. Thanks a lot. Thank you.